All right, let's get straight into it. Photosynthesis.
Let's start with the big question. We all kind of know plants need light to make their food, but how do you actually prove it? How can you measure the impact of changing light on how fast a plant photosynthesizes? Well, that's the exact problem this practical is designed to solve.
The core concepts. Look, it's so important to understand why we're doing this experiment. You can't just memorize a method, you've got to get the biology that's actually driving it.
It all begins with this equation right here, the fundamental recipe for how plants make food. They take in carbon dioxide and water, use light energy to power the reaction, and out comes glucose for food, and of course oxygen. Getting this balanced equation down is absolutely essential for your exam.
Now, this brings us to a really key idea, a limiting factor. Think of it like a factory assembly line. It doesn't matter how fast the rest of the line can go if you're waiting on one slow part. That one part limits the whole operation. For photosynthesis, that slow part could be light, CO2, or even temperature. If one of them is in short supply, it puts a speed limit on the whole process. And for this practical, we're deliberately making light our limiting factor.
Okay, let's get into the practical itself. Here's a step-by-step guide to the method you'll use to investigate light as that limiting factor.
So, here's our toolkit. We use pondweed, something like Elodea, because it photosynthesizes underwater and very handily releases little bubbles of oxygen that we can actually see and count. We've got a lamp as our light source, and the ruler is crucial for getting those distances spot on. The rest is pretty standard lab gear. Right, the key to this method is all about precision and making it a fair test. We set up our pondweed, pop the lamp at a set distance, and, this bit is really important, we let the plant get used to the conditions for a few minutes. Then we hit the stopwatch and count the bubbles that come off in 1 minute. We always do this a couple more times to calculate a mean. This makes our results more reliable and helps us spot any weird outliers. Then you just repeat that whole process, moving the lamp further away each time.
Now you've got to present it and, more importantly, figure out what it's all telling you.
A well-organized results table is your best friend. Seriously. See how it's laid out. Your independent variable, the thing you change, which is the distance, goes in the first column. Then you've got your columns for repeat readings and a final one for the mean. Keeping it neat and tidy like this just makes plotting your graph so much easier later on.
Now, when you plot your results, the number of bubbles against the distance, you're going to get a curve. You'll see that as the lamp gets further away, the rate of photosynthesis drops off. That's a great observation. But to get the top marks, you need to go deeper. What is the exact mathematical relationship here?
And the answer is something called the inverse square law. It sounds complicated, but it's not. As light travels from the lamp, it spreads out. So if you double the distance, the light has to cover 4 times the area, which means its intensity is only a quarter of what it was. That's why, to prove the relationship, you need to plot the rate against 1 over the distance squared. Do that and you should get a nice straight line going through the origin, which is the perfect way to show the link between light intensity and the rate of photosynthesis.
Right, let's talk exams. A huge evaluation point is the way we measure the oxygen. Counting bubbles is simple, yeah, but it's flawed. The bubbles aren't all the same size, and it's almost impossible to count them if the rate gets really fast. A massive improvement is to collect the oxygen in a gas syringe instead. This lets you measure the actual volume of gas, which is way more accurate and precise. Mentioning this shows you can really evaluate a method.
This is all about controlling your variables to make sure it's a fair test. Your lamp gives off heat, not just light, and that heat could mess with your results. So using a heat shield or, even better, an LED bulb that produces less heat is a great idea. And what if the plant uses up all the carbon dioxide in the water? Well, adding a bit of sodium hydrogencarbonate fixes that.
And here it is, the classic 6-mark question. Just think about everything we've just covered. You'd need to describe the setup, say what you're changing, the distance, what you're measuring, bubbles per minute, and what you're keeping the same, like temperature and CO2. And of course, mentioning repeats and calculating a mean will get you easy marks. Structure your answer logically and you'll nail it.
We have covered a lot, so let's quickly recap the absolute essentials, the things you really need to take away from this.
Okay, try to lock these 4 points into your brain. Number 1, the balanced symbol equation. You've just got to know it. 2, light intensity and the inverse square law. Remember that 1 over d squared trick. 3, control your variables. Temperature and CO2 are the big ones here. And 4, for those top marks, always be ready to evaluate the method. Talk about things like bubble size and how a gas syringe is better. And to finish off, here's a little challenge for you.
We've mastered light intensity, but what about light quality? How would you adapt this whole experiment to figure out which color of light plants love the most for photosynthesis? Have a think about what you'd change and what you'd have to keep the same. That's how you really start to think like a scientist.